a few really good audience questions came in, and piggybacking off of that manufacturing discussion, do you see companies like yours developing it in-house, or do you see CDMOs becoming experts in this space?
That's a great question. I think that really depends on who's doing the developing. So at this point in time, the vast majority of entities developing these are, either small venture-backed biotechs or, labs that are coming out into, spinning out, into an accelerator or into an incubator. So there's not necessarily the in-house ability to do that, right? So CDMOs right now, I think, are very, very important. Will they stay that way? Always some, for sure. If these things start to show a lot of promise and they get scooped up by, the Sanofis and the Roches and the Boehringer Ingelheims and Novartises of the world, I think there might be a shift in that because a lot of those companies like to manage their own manufacturing. But for the time being, CDMOs are going to be critical, and not just for manufacturing, but also for qualification, validation, identification of appropriate assays, and for release and qualification, appropriate analytics. I mean, they're going to be critical at this point.
Sure. Okay. Thank you. Another thoughtful question came in. What is the dose that the patient needs, and is this single dose or daily dose? How often should you inject to patient?
Oh, that's a great question, and I don't know the answer. What I can say is that we have based dosing for our wounds on the empirical data that was seen in animal studies and also in some clinical investigator-sponsored studies that were done and published by our CSO and co-founder, Dr. Evangelos Badiavas, at University of Miami. So we don't know the perfect dose yet for sure. We have, initially it was created from the number of, EVs that were made by a certain number of stem cells that were found to be useful and efficacious. So we currently give 6 doses for our EV patients, 3 for our burn patients if they don't close before. And there is some data internally as to why 6 might be good for the EV patients, but frankly, most of them close in that treatment period and don't get all 6 doses. They might reopen later, but at which point they will be retreated if they're in the treatment period. But we don't have a great handle on that. It seems like the 6 that we're giving every few weeks is doing very well. But there's also, what do you do in the out periods? Like, what do you do a year later? Do you do it again? Or it's not really clear what needs to be done for that. There's a lot more work that needs to be done on dosing. I do think, based on our work, that dosing at some point is probably pretty valuable.
Sure. Okay. Thank you. Next up, what is the source of the stem cells that you extract the EVs from, blood or tissue?
They're strictly from bone marrow-derived mesenchymal stem cells. So neither blood nor tissue, really. It's we get bone marrow aspirates from donors and isolate from them.
Gotcha. Okay. We have a few more that I would love to get through. What is known about potential EVs' shelf lives? If it is part of an ointment?
That's a great question, and we've contemplated things like gels and ointments and things. We don't have an experience in-house with different formulations that include ointments. Ours is more of a aqueous suspension. But I will say that EVs on their own and exosomes are very stable. So we've gone out several years and found that, and others have as well, and shown quite a bit of stability. So while I don't know in the ointment formulation, I know that in the aqueous suspension formulation, there's a lot of stability for years. So I wouldn't think that the stability would be altered dramatically by putting in an ointment. One thing that might be more challenging, as you might guess, is getting the EVs to get to the cells and be able to fuse and dump their cargo. I just don't know what an ointment type formulation would do to that dynamic. I think that might potentially cause a problem, but that would have to be looked at.
Sure. Okay. Are extraction purification strategies indication specific? Is your extraction purification strategy proprietary?
So the answer to the second question is yes, but are they indication specific? No, no, they're not. They're the same. So basically, the product is the same regardless of whatever. So in our graft versus host, burn, and EVs, well, they were different lots. They were all extracted and purified the same.
Gotcha. Okay. We had a question come in as during our registration process. Registrants are able to submit questions, which are often very good. And before we wrap up, I'd love to ask you, uh, we're curious how you approached release criteria, especially when the product is a complex mixture. What was the most challenging in establishing those specs?
Yeah, so that's a tough question all around. I think that so as I mentioned a bit before, um, our approach was to focus on the similar release characteristics of stem cells, right? So there are known profiles that are acceptable for stem cells. So some of those are compendial, many are not. So they have to be developed and qualified and then eventually validated. And then we focused, the hardest, as I said, is potency, and we focused on indication for that. And as I said before, that's not a great strategy for a group with a really big, diverse pipeline, because then you're going to have all these different potency assays and sets in the at least in the FDA paradigm. It's not one assay typically; it's a set of assays. So that can get to be a lot. You use a lot of, it's expensive. It's time consuming, and you also use a lot of your product. So you want to minimize that, right? That's the key thing, is how little can we use is the question you should be asking, because you are limited by how much you can make, and you don't want to use all of that in doing stability and release testing, et cetera. So we focus on indication, right? We said, well, what are the typical potency assays for wound products, and went with that. But that's, as we move into graft versus host, we're working on, well, now we don't have an appropriate, so now we have to figure out a new one, and that's something that we've got some work on. So yeah, it's not simple, but I think if you just go to kind of first principles of, well, if they come from the cells, what would you do if it was just the cells, and then go from there. And let's face it, the agencies don't know either, right? So it's kind of an open path to forge your own way. And as long as you can justify it and show that, hopefully, at least for potency, it's stability indicating, at least one or two of those, then it's likely that they'll at least give you, and in the early stage, they'll give you that leeway. As you get to sort of pivotal phase 3 stage, it gets harder. So it's just a challenge.